Good afternoon. Welcome to the Walls Lecture. This is the Florence Mahoney Memorial Lecture. Just briefly, for those of you who never had a chance to know Florence, she, throughout her full life of 103 years, was a strong advocate for NIH, for aging research, among other fields of study. But above all, she was a strong proponent for what she called real research, hard research, rigorous research. And every year when we were planning with her who the invited speaker was going to be, she took a very active role in making sure the science was up to her standards. And I know she would be absolutely delighted with this year's presentation by Dr. Ron DePinho. Ron has really played a unique role in convergence of science in the interest of elucidating basic mechanism to serve better understanding and ultimately translation. And the field of aging and the field of cancer research have long enjoyed well-recognized complementarity. After all, the essence of cancer is unconstrained growth. In the case of aging, some of the difficulties that arise with aging have to do with defects at times in cell growth and proliferation. There are common areas of metabolism, common concerns about genomic stability, and Ron has uniquely been at the forefront of research in both of these areas. He began his training at Fordham, stayed in New York through medical school at Einstein, some clinical training at Columbia, then was at Harvard, where he was a full professor since 1998, until very recently. So many of you will know that as of September 1st, he left Harvard, his professorship, and his role with the Belfer Institute there to move his program to a yet greater level of responsibility as president of MD Anderson, where he undoubtedly will be able to meet the challenges and fulfill all the capabilities of that august institution. Ron's work has taken a number of paths. It's used extremely well animal models to elucidate some of the basic issues of cell metabolism as they are relevant to cancer and to aging. He uncovered a great deal about the mechanisms of genes that are both protective or promoting for cancer. He was among the pioneers who understood the relevance of telomeres affecting the biology that again affects those two conditions. And most recently, and why it's particularly exciting to have him here this year, he has really elucidated the convergence of the mechanisms of tumor susceptibility, of telomere length, of telomere dysfunction with basic mechanisms of metabolic, in particular mitochondrial metabolism. Again, I'd emphasize a very unique product of an open and brilliant mind who has brought together areas of apparently, to many, disparate biology into what he'll present, I think, is a very compelling case for a new, very important integrated view of this biology. So, Ron, thank you for being here. It's a great pleasure to introduce you today.
Richard, thank you very much for that overly kind introduction. I understand that this is being simulcast to MD Anderson. Hi, y'all. I learned that y'all is actually singular. All y'all is plural. So I'm getting there. Anyway, it's just it's such a privilege to be here, and thanks again for that extremely generous introduction. I think the secret to my success has been having really phenomenal people that have come to my lab and just letting them cut loose, one of whom is in the third row there, Jane Stommel, one of your new stars here. So this is just a wonderful opportunity to celebrate some of the really cool science that's been going on in the lab and collaborators that we've been blessed to have. I'm going to be talking about the role of telomeres in cancer and aging, and what got us into this is really trying to understand its role in a variety of different biological processes: cancer, aging, degenerative diseases, both diseases that are acquired, like liver cirrhosis, which are the 7th leading cause of death by disease worldwide, and premature aging conditions such as ataxia-telangiectasia, Werner's, Bloom's, and in all of these processes, telomeres have figured prominently. Now, what got us interested in this space was trying to understand the intimate link between aging and cancer. Advancing age is by far the most important risk factor for the development of cancer. And interestingly, it's not all cancers. Cancers in aged individuals tend to be dominated by epithelial cancers. And those cancers also arise with very radically altered cytogenetic profiles that are characterized by complex non-reciprocal translocations and copy number alterations. And this is just an older ACGH tracing of a pancreas cancer showing lots of copy number gains and losses. And we now know from the Genome Project that therein resides oncogenes and tumor suppressor genes. So understanding the mechanisms that might drive why is it that age is such an important risk factor? Why do aged individuals develop the types of cancers they do? Why do those cancers have altered genomes? Are things that we've tried to answer. And this was further brought into focus when we looked at the mouse, which instead of developing epithelial cancers, had cancers that were more hematopoietic and mesenchymal in origin, and for the most part had an order of magnitude less genome complexity, typically whole chromosomal gains or losses, but at least tenfold less regional amplifications and deletions. And we felt that if we can understand the cross-species difference, we might illuminate the side of human biology that is about this epithelial dominance in the aged. And so for a number of reasons, we felt that telomeres might be at least one component of that. Telomeres are these structures that cap the ends of chromosomes. They help to maintain chromosomal integrity. In their absence, telomeres, the ends become recombinogenic. They also elicit DNA damage signaling, etc. In humans, telomeres are short and telomerase activity is low. And in the mouse, telomeres tend to be longer, especially inbred mice, and they have more promiscuous expression of telomerase. And so in a sense, mice do not experience one event in cell biology, which is telomere-based crisis. That as cells divide in the absence of telomerase, because of the end replication problem of DNA polymerase, you have the shortening of telomeres, and eventually you signal senescence via eroded telomeres, uncapped telomeres. So mice essentially do not experience telomere-based crisis. So we thought that if we can engineer mice to experience crisis, then we might understand better the role of telomeres and telomerase in aging and in cancer on the organismal level. And so to do that, we collaborated with Carol Greiter. Maria Blasco, then a postdoc in her lab, had just cloned the gene that encodes the template for the telomerase holoenzyme. It serves as a template for the de novo addition of telomere repeats. There's also a catalytic component, a reverse transcriptase. We've knocked out this, as I'll tell you in a moment. We've also generated knockouts for this gene, as others have, like Leah Harrington, and have studied them. And for the most part, they phenocopy one another, with telomere maintenance being the critical activity of this holoenzyme. And so when we knocked it out, and the we in this case is Hanun Lee, and in collaboration with Maria Blasco, who generated the mouse, and then Hanun went on to characterize the phenotype of the mouse. And essentially what we saw was that in the first generation knockouts, they were pretty healthy, and they had long telomeres. So we crossed them through successive generations to get them down to a more humanized telomere length. And by about the third generation, in collaboration with Peter Lansdorp, we began to see cytogenetic evidence of loss of telomere function. You see here these Robertsonian end-to-end fusions. There's no detectable telomere repeat at those fusions. So they've recombined consistent with the McClintock hypothesis established in the 30s. And coincident with the cytogenetic evidence that telomeres are becoming dysfunctional, Hanun Lee, Lienhard Rudolph, Kwok-Kim Wong went on to demonstrate that these animals have a premature aging condition, a diminished capacity to handle acute and chronic stress, just as aged individuals do. There was also widespread organ atrophy, especially organ systems with high proliferative histories like the GI tract, the testes, and the hematopoietic compartment, for example. And we now know that that was due to depletion of resident stem cells. Linda Chin, inspired by the work of Mike Kasten, went on to ask the question of whether or not p53 might be sensing telomere dysfunction. Mike had taught us that DNA damage activates p53 and activates in turn a cellular checkpoint response. And so she asked whether or not if telomeres might be an eroded DNA damaged, and perhaps p53 might sense it and execute these checkpoint responses. And indeed that turned out to be the case. So she brought p53 through these successive generations. And this is just one example of the impact on an organ system. This happens to be the testes of a 6th generation animal. And here this is a littermate that has p53 competent, where you see barren seminiferous tubules due to apoptotic elimination of the germ cells. And here you have restored the cellularity of the testes in this littermate control with p53 deficiency and about a 90% reduction in the amount of apoptosis. So now you have a situation in the organism where you have cells that are surviving and dividing at a time when the genome is becoming unstable. And what we saw was an increase in cancer. And this was a surprise to everybody in the field, because at that point the thinking was that telomerase was needed for the development of cancer. And it turns out to be the opposite. And this is the power of genetics and model systems. What we learned from this experiment is that it's the combination of telomere dysfunction coupled with a deactivated DNA damage response that enables one to create the large number of changes needed for the development of cancer, for cancer initiation. And so what was more striking in those studies is not only did we see more cancer, but the tumor spectrum shifted towards a more humanized spectrum dominated by epithelial cancers. Those cancers had altered cytogenetic profiles, non-reciprocal translocations, regional amplifications, deletions, and so on. And we think the basis for this relates to that fusion bridge breakage cycle where you have opposing kinetochore attachment of the fused chromosomes, cell division, and breakage of chromosomes as they move to opposite daughter cells. I should also point out that the genetic context, the genetic luggage that a cell carries on its way to crisis dictates how crisis will behave, either to promote or suppress the development of cancer. We did exactly the same set of studies in the INC-ARF model. Cancer-prone develops lymphomas, sarcomas, but it has an intact DNA damage response, and there was no change in the tumor spectrum. So what these studies taught us is that the combination of telomere dysfunction and p53 inactivation allows us to acquire the large number of changes needed for epithelial carcinogenesis. In a sense, mice are protected from epithelial carcinogenesis because they're missing this mutator mechanism. Now, one of the things that we also did was to compare genomes. As you can tell, this is an early platform for array CGH. But the bottom line is that there was at least a 10-fold increase in the amount of amplifications, deletions. And we looked across multiple tumors. We found that recurrent amplifications were targeting oncogenes that were important in the pathogenesis of that cancer. This happens to be a colon cancer. We see amplification of KRAS. In a breast cancer, we saw amplification in 33% of samples for HER2/neu, so on and so forth. And so we think that telomeres are particularly important in providing us with this explanation as to why age and cancer are intimately linked, certainly not the only mechanism, but we think it's an important one, why you develop the types of cancer you do, and how those genomes are shaped. So you have a lifetime of epithelial renewal, and it's important to appreciate that you turn over your GI tract lining every week, your skin every 2 weeks. You create a trillion blood cells per day. There's an enormous amount of renewal that goes on. So you essentially walk this telomere plank. If you deactivate the p53, and also further accelerate with conditions that are pro-inflammatory conditions, high ROS conditions, etc., those will further fuel erosion and generate cancer-prone conditions. And you have this fusion bridge breakage. And Jeff Wall taught us that when you break chromosomes, that provides a nidus for amplification and deletion at the site of break. And we think that that then creates an opportunity for all wholesale genome changes up front. At the benign to malignant transition, there's an explosive onset of genome change that occurs. And we think that telomere dysfunction plays a major role in epithelial carcinogenesis. And that allows you to initiate these cancers. So telomere dysfunction is important for cancer initiation. But one of the things that we noticed very early on in these cancers is that they had a very feeble progression phenotype. They were poorly invasive, non-metastatic. They were hard to adapt to culture and propagate. And so we speculated that perhaps while telomere dysfunction might be important for initiation, telomerase may be important for progression. How it's playing a role in progression was unclear. Whether it's further checkpoint responses that need to be overcome, or whether it's too much genomic instability that's even unhealthy for cancer cells that needs to be quelled, we didn't know, and we speculated, but the experiment had yet to be done to assess the impact of telomerase reactivation after having gone through crisis. So I'll tell you about today is a set of experiments that we've done where we've engineered a system that enables us to ask the question of what is the role of telomerase in cancer progression. And this is an inducible system, so it also affords the opportunity to extinguish telomerase in a fully formed tumor in vivo so that we can assess the impact of telomerase extinction and the adaptive responses using genome scale analysis. So before I tell you about that study of telomerase activation and extinction, I want to just go through a published set of data that was done by Ergin Sahin, a really gifted postdoc in the lab, to understand the telomere checkpoint response. And this is in normal cells. We had shown years ago that P53 is important, as I mentioned, and that it executes the classical cellular checkpoint responses that leads to organ atrophy. Well, that makes it's a good explanation for tissues with high proliferative histories, such as the GI tract, but it's not a very good explanation for the kinds of other pathologies that we're seeing in these late generation animals, which we saw things like cardiomyopathy, which was first described by Anna Versa and Blasco years ago. You see this echocardiogram with large dilated left ventricles compared to the normal, very thin ventricular walls, decreased fractional shortening, and so on, very sick hearts. And also you see an impaired metabolic profile. These animals have a very poor response to fasting, so they can't mount a robust gluconeogenic response, suggesting that somehow the liver is affected. And these are not very highly proliferative tissues. So how do we rationalize these phenotypes in the context of telomere dysfunction? So we took an approach that was unbiased, and what Ergin did was to look at multiple organ systems with different proliferative capacity, such as hematopoietic stem cells, the liver, which is relatively quiescent, but has the capacity to renew, the heart, and we've looked since at several other tissues as well. And in the context of telomere dysfunction, we asked what networks were perturbed in the context of telomere dysfunction. That was a common theme across all of these tissues, to see if we had missed something. So we saw the typical P53 checkpoint responses, et cetera, and that network being activated, apoptosis and so on. But one consistent observation was these pathways that were intimately linked to mitochondrial biology. This was even noticed years ago in a yeast study, and the assumption was that the investigators were not feeding their yeast well enough, and that there was somehow a problem, an experimental problem, and the focus was on other pathways in the context of yeast. But essentially the same profile was secured over a very large phylogenetic distance and in mammal systems across multiple tissue compartments. In addition to the mitochondrial dysfunction on the gene level, these are microarray analyses, we saw that virtually every complex of the mitochondria had repression of genes that are important for ETS function. And this was confirmed by Ergin by RT-PCR. So it told us, and in some cases Western, it told us that telomere dysfunction is somehow linked to mitochondrial biology in a variety of different tissue contexts. And that got us thinking, because we knew of the work of Larry Donauer in P53, Larson with POLG, Bruce Spiegelman with PGCs, that a lot of these are knockouts. These genetic model systems have very common phenotypes of degeneration across these systems. So you have defective mitochondria due to impaired DNA synthesis and proofreading. You have the master regulators of mitochondrial biogenesis. They make mitochondria, PGCs, and they make mitochondria work because they regulate almost all the genes that are important for mitochondrial biology. Also very important in oxidative defense. and of course the work of Larry Donauer with germline activation of p53. And we had shown when you knock out p53, you can attenuate a lot of these phenotypes. So we had a convergence of phenotypes. The question is, are they linked? So the first thing we did was to look at the PGCs, and we found, in fact, that PGC1 alpha and beta, the two key regulators of mitochondrial biology, and the G4s, the late generation, were significantly decreased relative to wild type or the G1s. And these are just a few of their key targets, like TFAM, important for DNA synthesis in mitochondria. Those were also coordinately repressed. So this told us that telomere dysfunction is somehow repressing PGC and is potentially causing the kinds of problems that we're seeing. So we wanted to actually ascertain whether or not mitochondria were in fact compromised. And so reducing about 7 years of work to one slide, just showing a few snippets. This is truly like centrifuging down a lot of data and giving you the pellet, ultra centrifuge. What he saw was in these tissues that mitochondrial DNA copy number relative to nuclear DNA is decreased in the G4s, that at the level of EM, stereotactically, he was able to show that there are fewer mitochondria, that he looked at all the complexes, just showing you complex 1 and complex 4 were decreased in their activity. They had a profound decrease in the ability to generate ATP and a very significant decrease in state 3 respiration, ADP-stimulated state 3 respiration. And for the aficionados, this is on not just a whole lysate, but this is on a per mitochondrial basis. So you have fewer mitochondria, and the mitochondria that you do have are less functional. So then the question is, what connects to PGC? And an obvious candidate was p53, which typically is thought to activate genes, but on occasion has been shown to repress genes. It was quite possible that there was an intermediate step here. But nevertheless, we looked at p53, and we first looked at the genetics. We wanted to ascertain whether or not there was a reversal in the transcriptomic impact of telomere dysfunction on the PGC network. And so what we show here is the decrease in mitochondrial DNA copy number with a rescue when you have p53 deficiency. You have a decrease in PGC1 alpha with a partial rescue, PGC1 beta complete rescue, and the downstream targets a significant rescue in these various targets. So at least at the mitochondrial level and the transcriptomic level, when you knock out p53, you alleviate what appears to be repression on PGC. I'm not going to go through all the data, but he then did in silico analysis of the promoters, identified 3 evolutionarily conserved binding elements predicted to bind p53, 2 in the PGC1 beta promoter region, and this is the positive control of chipping p21. All 3 of these identified cis elements were able to pull down with p53 antibody, and the 2 that were identified in PGC1 beta also came down in this experiment. And these are just reporter assays that I won't go through, but essentially it shows that when you knock out p53, you alleviate the repression if you use those cis elements in the reporter. So again, I'm not going to go through the enormous amount of data that Ergen did, but at each step along the way, he secured genetic and biochemical evidence for this circuit. For example, in the animal, he used adenovirus TERT to see if he could rescue the metabolic defect in the liver. And that was accomplished, in the sense that when you put TERT back in, you deactivate p53, rescue PGC, and rescue the metabolic compromise. So this ruled out that there might be some defect in the gut, and the metabolic defect is because of some other organismal process that we're missing. So it shows clearly, and you also isolated hepatocytes to prove the same thing. I had just shown you that when you knock out p53, you rescue the transcriptomic repression, and indeed this is just a phenotype showing that you rescue the cardiac function in this particular experiment when you are deficient for p53. So this is now in a telomere dysfunction, knock out p53, and you rescue the impact that you have on the heart. I just showed you the direct target data. And the last thing that he did was to use adeno-PGC1 alpha, again in the liver and isolated hepatocytes, to show that he could rescue the gluconeogenesis. So this is a very clear-cut pathway that has genetic and biochemical and genomic data that clearly establish this as a key pathway in the response to telomere dysfunction. And in looking at how we now think about the aging process, and Richard alluded to this, there's the thinking that you have, these different processes that somehow are not that connected, genotoxic stress that might contribute to aging biology, or mitochondrial dysfunction, etc., that would further fuel compromise of the organism as a function of age. I think that they can all be integrated with some of the observations that we've made. First and foremost, with respect to telomere dysfunction, p53 continues to play a very important role by doing its classical functions of activating genes that cause apoptosis, senescence, and cell cycle arrest. In addition, we now know that p53 directly links to PGCs, and PGCs not only regulate mitochondria, but they directly regulate many genes that are involved in oxidative defense. And that's important because when you have mitochondrial compromise and less flux through ETS, you actually generate more ROS. And at the same time, you're decreasing your ability to defend against the ROS. So it's a very strategically poor thing for the cell to do to repress PGCs, given its role in these two processes. Moreover, when we think about ROS itself, any condition that creates ROS, it's particularly injurious, obviously, for the genome, but in particular for telomeres, which are G-rich, and ROS loves to attack G-rich sequences. So you can imagine that this might create a feed-forward loop and provide an explanation for an observation that's been long seen, which is that we do very well for decades, and then in that last decade of life, those last few years of life, you really have this accelerated decline in your physiological reserve and robustness, capacity to respond to acute and chronic stresses. And one could imagine that that might relate to the fact that over a lifetime you accumulate damage, telomeres don't repair the damage, they serve as a reservoir, you eventually tonically signal to p53, you start in motion this cascade of events, increase intracellular ROS levels that would then be injurious, and you then create more damage, and you continue the cycle still further. So this is a pathway that as we look at from the aging standpoint, if you think about many of the genetic elements that have been discovered in aging genetically, such as the sirtuins, components of the PI3 kinase pathway, now that we know this core pathway, many things directly linked to modulation of flux through that pathway. So we think this is a core pathway, a very important pathway for aging, certainly not the only one, but a critical one. If you just think about the sirtuins, sirtuins modify p53 and PGC. They repress p53 activity, and they activate PGC. Hence, they're very good points of intervention from the standpoint of attenuating the aging process as this pathway is concerned. So I think that this is going to be an opportunity for us to provide a framework for how to integrate a lot of the things that we've known about aging, mitochondrial biology, genotoxic stress, viewed through the lens of this axis. So now let me tell you about the story of modulating telomerase and the response of cancer cells to flipping the switch on and off. This is a lot of work that a postdoc in the lab did, Mariella Jaskelioff, where she knocked in the estrogen receptor into the open reading frame of the telomerase reverse transcriptase to generate a fusion protein, TERT-ER. This moiety is responsive to tamoxifen, such that when you add tamoxifen, you release from HSP90, you renature the protein, and the protein can now act on its substrate. This has been championed by Gerard Evan and others. It's a very effective way of regulating proteins at the endogenous level for activity in the cell. And this then, with tamoxifen, allows the telomerase to act on its ends. And it works like a charm. I'm not going to go through all the data, but this is a vehicle-treated G4 cells that were clearly eroded for their telomeres. You see very little ends here. And this is after a bit of treatment with OHT, which now restores the ends of the telomeres. So Jan Hugh in the lab was interested in asking the question if he could exploit this system to toggle telomerase on and off at specific points in the development of cancer. And he turned to the ATM model. This is an allele that was generated by Fred Alt's lab. It's a lymphoma-prone condition. They generate lymphomas and are dead more or less by a year of age. And he crossed that on to the telomerase knock-in, TERT-ER, and then brought them through successive generations. And I'll tell you about the wild types and the G1s are pretty similar. They have ample telomere reserve, and the G3s and G4s in this particular experiment have severe telomere dysfunction. And as we had shown with this straight conventional knockout for telomerase against the ATM knockout, what you see when you have telomere dysfunction is that the animals live longer. And the reason for that, even though it's ATM deficiency, you still have an intact p53 checkpoint response, and Carol Greider has shown the same thing in her lab. So what you have is a elimination or a decrease of the number of cells that are able to become malignant and eventually kill the animal. So you see a prolongation in survival. And here, what you see is G1 with or without OHT. So the presence or absence of telomerase activity has no impact on the kinetics of the tumor and the survival of the animals. And here what you see is when you add OHT at about 20 weeks, where we know that there's initiated cancers in the thymus already, that you have, in fact, an acceleration in the death curve here. So suggesting that if you go through crisis and then reactivate telomerase, that you have now a more aggressive cancer, relative to even those that had intact telomeres all along. So this process, this biphasic process of crisis coupled with telomerase activation is providing new biological capabilities for these cancer cells. And that on many levels they're obvious. You could see that the control animals, the thymus, had cancer, but these are very aggressive malignancies. We've deactivated Andre Nussensweig's favorite protein, 53BP1, also decreased senescence, quelled p53 signaling. And as I mentioned, these are aggressive cancers and they take on new biological capabilities where we have, for example, in the long telomere animals, they never go to brain, whereas in the context of having gone through crisis and reactivating telomerase, we see widespread aggressive involvement of many different organ systems, such as the brain, etc. So I think that this is acquiring new genomic events through this mutational mechanism that endows these cells with increased biological capabilities. And that's clearly the case in terms of looking at the genomes. We see the red bars are the ones that had telomere dysfunction that had been treated with OHT, and these have telomere intact, and they're both ATM deficient. And these lymphomas, the red bars are the ones that had gone through crisis and reactivated telomerase. And they acquire signature events that lead to amplification of all of the genes we know to be important in lymphomagenesis, such as some of them listed here, like Notch, FLT3, MYC, deletions in P10, etc. And we don't yet have enough samples. These are very limited sample sets for us to be able to ask, well, what might be different in the telomere intact versus the ones that had gone through crisis and then telomerase reactivation, to say what's conferring the aggressive behavior in those cancers. So we have to increase the numbers yet. So then we asked the question, having activated telomerase and generating these tumors, what would happen if we were to flip the switch off? What are the adaptive responses? And this is important because in clinical trials, there are agents that are directed against telomerase. So what we did here was to take one of these late generation cell lines, tumors, I should say. They didn't see plastic. They were passaged immediately in through successive generations, successive passages. And they were left on, so this is telomerase on, so that's the control arm. And then another arm that we removed OHT and passaged, and these were telomerase off. And I'll tell you that basically around P2, P3, depending on, we did multiple different cell lines, depending on the cell line, they start to enter crisis around here, and then they reacquire their aggressive tumor capabilities, having still turned off telomerase, and they have interesting adaptive responses. So this is P1, the Kaplan-Meier curve of with or without OHT. Not much happens. In this particular cell line, you can see now when in the vehicle-treated telomerase off, they start to, more of the animals survive, the cancers come on later, and yet they then reacquire their tumorigenic capability. So what are the adaptive mechanisms? This is just showing you that you got progressive telomere shortening and then acquisition suddenly of longer telomeres with a wider base suggesting heterogeneity in telomere lengths. So we also saw that in around P2, we saw activation of the P53 checkpoint response, 53BP signaling, increased apoptosis, increased H2AX. Now it's important to appreciate that you don't quite rescue all the way. When you adapt, you do rescue apoptosis completely and cell cycle arrest. But with respect to genotoxic stress and signaling, there still appears to be, you don't return to wild type. So there's ongoing genotoxic stress here, and that's important to keep in mind. So obviously this is a pattern that might be consistent with ALT. This is a telomerase-independent recombination-mediated mechanism discovered by Roger Riddell that can help maintain telomeres, albeit inefficiently. The hallmark features of ALT is heterogeneity of telomere lengths, extra chromosomal fragments, ALT-associated PML bodies, and higher levels of sister chromatid exchange. I'm just going to summarize some of these data here. So this is what the telomeres look like when you keep telomerase on. It's maintained. And then as you go through these passages, you see that you start to lose signal, and then suddenly you reacquire signal. You see increased extra chromosomal fragments that are quantified here, increased APBs quantified here, and increased sister chromatid exchange at telomeres, as you can see here from these different colored dots here. And that's significantly increased as well. So those are classic features. So clearly, at least one adaptive mechanism is what we would have expected, which is the acquisition of this recombination-mediated ALT mechanism. But we wanted to ask whether or not there were additional adaptive responses, and so we took a genome-wide approach to transcriptomic analysis of the controlled telomerase versus the telomerase off ALT cells and the parental cells. We actually went through a series of controls where we filtered out the OHT responsive genes, and I won't go through those details, but again, what we see is a common theme. The number one network that gets activated are things that are connected to mitochondria. The things that get downregulated are things that are involved, let's say P53 signaling, apoptosis, things of that nature. But the upregulated ones seem to suggest that one of the things that these cells want to do in the absence of telomerase is to try to support mitochondrial function. And in addition, there is, in the genes that were upregulated robustly in the ALT cells, so this is telomerase, this is wild type, the P0 parental, these are the ones that were maintained, second bar on telomerase, and the last third bar are the ALT cells that broke through. And you could see that PGC1 beta, the master regulator of mitochondrial biogenesis, and a lot of these genes involved in oxidative defense are being upregulated. And also they had higher levels of ROS. Interestingly, in these tumors, now what's great about the telomerase knockout is you get amplifications and deletions, and under biological pressure they select for oncogenes and tumor suppressor genes. So we can ask, are there new amplifications that occur or deletions in these tumors? And the one that really matched nicely with overexpression of PGC was amplification of PGC1 beta. So it's overexpressed, and there is a genetic pressure to amplify that locus. And Jan then went on to look, with the help from Ergen, at mitochondrial function. So as cells are going through crisis, mitochondrial function is significantly decreased. And then you break through to ALT, and mitochondrial function is restored, but not to wild-type levels. And we see that ROS levels, which go through the roof in the cells going through crisis, are significantly alleviated, but again, not completely so in the context of ALT. So we asked the question, if the cells are trying to adapt by maintaining mitochondria, if we were to knock down PGC1 beta to physiological levels, what would happen? Or oxidative defense genes. And so we did this for SOD2 as well as for PGC1 beta. And when you knock it out in the telomerase positive cells, they don't quite do as well, but there is not a significant impact, and those animals all still die. However, if you knock out in the ALT cells, PGC1 beta, knocked down, I should say, what you see is a lot of the animals being cured of their disease and a significant prolongation in the onset of tumor formation. And the same was true for SOD. If you do this in cell culture-based systems, it's a shift, IC50 shift of about tenfold. So it clearly is an important adaptive response. So what we've learned over the years is that telomeres play important roles in apoptosis, senescence, and mitochondrial biology. That when you have telomere dysfunction, you activate apoptosis, senescence, and compromise mitochondrial biology. In the context of ALT, you eliminate these checkpoint responses, guided mostly by p53, and you do have some restoration of mitochondrial function and oxidative defense, but not at sufficient levels to really maintain robust growth and development of these cells. And they try to maintain this axis through upregulation of PGC1 beta. Now, one thing I didn't mention is that we also saw amplification in recombination genes involved in homologous recombination. So the MRN complex is thought to be very important for ALT and also the Fanconi proteins, and a few of those were upregulated. I think it was MRE11 was amplified, and one of the FANC proteins were also amplified. So what we've learned is that telomerase plays an important role in aging and degenerative disease, that dysfunction activates p53, which activates cellular checkpoint responses, represses PGC, and impacts on mitochondrial biology and oxidative defense. And in the context of cancer, telomere dysfunction is important for cancer initiation, and in the context of p53 deficiency, enables you to acquire a lot of genome-promoting alterations that enable epithelial carcinogenesis, and that telomerase activation plays a role in cancer progression. And it's the combination of crisis and activation that enables cells to acquire more biological capabilities for full malignant transformation. And that its extinction can be countered by adaptive responses that involve recombination-based ALT as well as mitochondrial and oxidative defense. So in the last few minutes, I want to just describe one last set of experiments that describe an attempt to also exploit this inducible system to understand whether or not processes of aging, degenerative conditions, can be modulated. There's a lot of activity in which pathways that are known to be integral to the aging process are being modulated, the PI3 kinase pathway, et cetera, and it's showing very interesting results in terms of lifespan regulation and attenuation of age-related disease. And so we're interested, given the prominence of telomeres in precipitating age-related disease, whether or not its regulation experimentally could alleviate some of the degenerative conditions that I talked about. So what we did was, first of all, to show that this TERT-ER inducible system recapitulates the phenotype of the conventional knockout. It is identical to what we had seen previously. This Kaplan-Meier curve shows that these animals die sooner, and they have all of the pathological sequelae that we see with the conventional knockout. I'm not going to go through all that data. Taking ear fibroblasts and putting them into culture with or without OHT, and this is the work of Mariella Jaskelioff, you see that in the presence of OHT, the cultures take off, and then these that are in vehicle maintain undergo a senescent response. And then if you add OHT, these cultures that are significantly decreased in their proliferative capacity have the ability to now reacquire growth. And you can see here that you deactivate some of the p53 checkpoint responses. On the molecular level, p21 goes down, MDM2 goes down, p53 goes down, and senescent, and these cultures really take off. On the organismal level, if you look in the GI tract, you see lots of apoptosis when you have telomere dysfunction. If you treat the animals for 1 month with OHT, and then 1 month after termination of treatment, you look. So all the experiments I'll tell you about are after that 1 month. What you see is an alleviation of the apoptosis. You also see that testes size, which goes down, increases. Fecundity, which goes down, increases. This is a remarkable result where these animals that are largely infertile restore their fertility after OHT treatment. So these are these barren seminiferous tubules in a classic telomere dysfunctional. This is after a month of treatment with OHT, you restore the cellularity of the testes. The organ system that we focused on the most was the brain. Brain is really central to a lot of the problems that we experience as we age, so we wanted to really understand the impact on the CNS. And I think you can appreciate that these brains are much smaller in the context of the late generation, but if you add OHT, these brains can go back to near normal size, and this is just quantified here. Now, one of the main things that happens in the brain as we age is you get decreased myelination. And one of the main things that we saw were decreased oligodendrocytes, OLIG2 positive cells. And those cells that were there were less mature. They had less ability to generate myelin. But as soon as you add, so you see that these are the controls, the telomere intact. This is what the late generation looks like with OLIG2 positive. And then you add OHT, and you see that there's a dramatic increase in the number of OLIG2 positive cells. In addition, you restore the G ratios, the ratio of the amount of myelin sheath relative to the axon diameter, which is more or less in sync. And what you see is that the G ratios, that is, the myelin gets much thinner in the context of the G4 vehicle treated, but OHT, you actually restore myelination in these animals. Now, we looked in vivo, wanted to show that pharmacodynamically we're actually restoring telomeres in the brain. So we looked at the corpus callosum, the striatum, and the subventricular zone, and in each case, we're able to show that telomere reserves increased through in situ methods. And we're particularly interested in the SVC because that's a region in the mouse brain where stem cells reside, where new neurons are generated. In the SVC, those neurons go out rostrally to generate interneurons to maintain olfaction, innate responses throughout life, and they generate new interneurons in the olfactory bulbs. So we're particularly interested in this compartment. The first thing we did was in vitro studies. We went and took out SVC neurospheres. Again, the we in this case is Jihei Paik and Mariella Jaskelioff. And what they found in these neurospheres is that there's significant signaling, DNA damage signaling in the G4, and that the addition of OHT quelled the DNA damage signaling. When we tried to generate successive neurosphere cultures, we were essentially unable to generate tertiary neurospheres. and yet when we added OHT, we were able to regenerate the renewal capacity of these neurosphere cultures. We had shown years ago that when you have telomere dysfunction, you significantly impact on the ability of neural stem cells to generate TUJ-positive neurons. And here we see that that is significantly decreased relative to controls, and that if you knock out p53, you can restore that. But if you just add OHT, you can significantly increase the number of neurons that you get out of the neurosphere cultures. We then looked in vivo in the brain in the subventricular zone. So you see proliferative activity. When you have telomere dysfunction, that is significantly decreased, and you restore proliferative activity when you add OHT. Markers of stem and progenitor cells, such as SOX2, are robustly expressed in the subventricular zone and telomere intact, decreased in telomere dysfunctional, restored with OHT. We have DX double cortin positive cells. Those are markers of newborn neurons. And what you see is a significantly decrease in the number of newborn neurons and a restoration of the double cortin positive cells with the addition of OHT. So the question is, these newborn neurons, which eventually go out to the olfactory bulb, are they really generating renewal capacity for the brain on a physiological level? And to do that, what Mariella did in collaboration with the Flyer Lab was to do a well-known test, which is you blot either water or a noxious odor, such as 2MB, and look where the animal is in the cage. So this is an innate response to spoiled food or predator, where the animal with a noxious odor will go as far away from the odor as possible. It's not a learned response. And we did different concentrations. So the animals with water will traipse throughout the cage. If they're wild type, they will go to one side. And if they're knockout, they tend to go into all quadrants. Although they do have some function, they do tend to stay away from the noxious odor. And this is just quantified. At lower doses, you see that there's very poor discrimination that the telomerase deficient animals have with vehicle control. These are the wild type animals, G0, telomere intact. And with the addition of OHT to this cohort, we now restore the ability to discriminate these noxious odors. So these newborn neurons are not only getting generated, but they're migrating out, they're generating new interneurons and restoring this innate physiological function. The last slide is just lifespan of these animals, significantly decreased if you just treat them with vehicle. Just one pulse for 1 month is able to significantly restore the robustness of these animals, and they live a bit longer. Not to the level of the wild-type animals, though. And one thing that we can discuss is the cancer spectrum of these animals. They did not develop an increase in cancer. So just to summarize, I didn't show you data, but telomeres play very important rate-limiting roles in the pathogenesis of premature aging conditions. If you knock out Werner's in the mouse, you get no phenotype, work from Leader and others, and Garante. If you layer on top limiting telomeres in the mouse, you then precipitate classic features of Werner's. This is telling us that telomeres are rate-limiting pathogenic elements in those and other diseases. In the context of normal aging, I think it remains to be determined whether or not telomeres are playing an important role, and if we were to repair telomeres, whether or not that would impact on normal biological aging. But in the experiment that we did do, what it does teach us is that there is a point of return for tissues that are in a very severe state of degeneration. Recall the testes and how atrophic it was. Tissues, even in a very severe state of degeneration, retain a remarkable capacity to renew themselves. So as we understand the circuitry of aging, which I consider to be a disease, and pharmacologically manipulate those pathways in a way that's safe but appropriate for age-related pathology, we have a chance of impacting on normal aging, but even if we didn't go as far as to really impact on just the normal aging process alone, it's pretty well established at this point that telomeres correlate with age-related diseases. Over the age of 60, there's a very significant increase in Alzheimer's, cardiomyopathy, diabetes, and cancer if you have shorter telomeres. And so this is telling us that this is at least part of the process on a population-wide level. And the reason why this is important to appreciate is the impact of the great diseases that we're going to be facing. You know, when I was born in 1955, the average life expectancy in China was 42, and today it's 74. After the age of 60, for Alzheimer's, you have a near doubling every 5 years of the incidence of Alzheimer's, so that over 40% of individuals by the age of 85 have dementia, have Alzheimer's. And by the year 2025, there are going to be 1.2 billion individuals over the age of 60. And so this, you know, our world is careening towards an enormous challenge because of the changing demographics, where I think what's going to dominate politics, economics, societal issues is going to be our aging population. That is going to be the great challenge that we're going to face in this century. And, you know, while managing healthcare is very important, optimizing, having better evidence-based strategies to quell and manage patients with the disease, there is only one answer, and it's science. We have to figure out the pathogenesis of aging and how aging relates to these great diseases, because if we do not, we have a non-sustainable situation. And 2025, that's only 14 years from now, we're going to have over 1 billion people that are going to be acquiring these diseases. So I hope that our elected officials think beyond the election cycle and support science to the level that humanity deserves. And this great campus, the great state of Texas, and the rest of the world can make a difference if the wind is put in our sails. So let me just thank you for the people that did the work. We have Jan Hu, who did the TERT-ER cancer study, Maria Jaskelioff, the reversal of aging, Ergen Sahin, who has done a phenomenal body of work in discovering this pathway that links genotoxic stress to aging. We are really, these are people on the left here are individuals that really were, you know, on the Nina, the Santa Maria, and the Pinta, trying to figure out, you know, whether or not telomeres are important, and all of our collaborators, and just really grateful for all of the gifted colleagues that I had a chance to interact with at Dana-Farber and Harvard Medical School. It's a phenomenal environment, great trainees, just great colleagues, tremendous amount of talent, and I'm just inspired by the level of science and the work and the chance to do great good at MD Anderson. There's 18,000 people, an enormous amount of resources, a singular focus on cancer. There's a real chance that we together can do great things and really do what people are expecting us to do, which is to bend the curve on these diseases, if not cure them. Thank you very much.
Ron, that was great. If people want to use the microphones for questions. Let me start with one. As intriguing and impressive as the synthesis of pathways was that linked genomic stability, DNA damage, mitochondrial metabolism, to your closing challenges of using this information to identify interventions to modify aging to the good, the challenges also seem to increase with the complexity of the systems, and that perturbations and the predicted outcomes are not so simple. Do you want to comment on what you think the approaches need to be to that?
I think it's the same thing in cancer, where you look at mTOR, it's immunosuppressive, yet it attenuates signaling, and you get some pretty good responses at the organismal level and so on. You have to understand the details of how these things are regulated, right? It's the context, and it's exactly where to intervene. You can imagine that if you were to do an agonist for PGC, that you'd have problems with, gluconeogenesis and so on. That might not be useful. If you overexpress it too much, you might actually get mitochondrial proliferation. That would not be useful. So obviously you need to understand the target. One thing that I think would be interesting to consider would be if you could reawaken telomerase, because it is an important element, early enough in life, before you get into the initiated cancer stage, one could imagine that would be helpful. Or find ways to enhance DNA repair activity. Or something that would modify signaling flux through that pathway. We think in those terms in the PI3 kinase pathway, but we don't tend to think of it with the P53 PGC axis. So now that we have that axis, we can think, are there modifiers that can have the right kind of signaling attenuation so that we can dampen the response, so we can make more mitochondria and make them work better, or increase oxidative defense. So, I think the sirtuins are a good example of that, where you have this, ability to kind of regulate P53 in a certain way and PGC in the right way as well. So those are the kinds of things that if we think about and set things up are important. The challenge there is that, and this is, I think, a key point that we've learned, is that cell culture-based systems are not good systems in which to study this axis. This is a very important point, because we're growing our cells in Snickers bars conditions, a hyperoxic state, and they're not as dependent on mitochondria, right? They're very glycolytic. And so you don't see so much dependence on mitochondria. So as a result, if you're studying mitochondria in like senescence or aging, you may not get the full story. So I think, not to say that they're not useful, that model system. All model systems have advantages and disadvantages. But if you're going to be looking for screens, you got to know what it is to look for and what context to study that perturbin so that you can get the right answer.
Thank you very much. That's very elegant work that you did there, and that 7-year length scale is about the same place that we're at, so that's encouraging. My question actually has to do with the fewer mitochondria that you observed, and that's, if you could comment, is it an issue of, to be simplistic, poor synthesis of the mitochondria or higher depletion? Are you seeing anything in the chaperone proteins or the HSPs that are showing something like that?
Yeah, it's a very, very good question. We haven't looked. We're very interested in looking at mitochondrial, recycling and so on. We don't know. But it's very important. TFAM is significantly decreased, so mitochondrial DNA synthesis is down. So, clearly that's one aspect of it. But we can't say. But we do know minimally on a per mitochondrial basis, they're not working well. But I think there's a very interesting biology over a large phylogenetic distance that relates to how mitochondria are maintained, renewed, and so on, that we should look at, but we haven't.
Very, very interesting lecture. My interest is the association between chronic inflammation, aging, and cancer. The aspects of telomerase association with apoptosis and its reversal by ALT that you mentioned, including the superoxide dismutase 2 from mitochondria, signals to me that there is an imbalance between the balance that should be to keep immune surveillance of the tissue, to maintain immune surveillance. And it's an imbalance between apoptosis and wound healing processes or growth arresting and growth promoting events that is going on in a normal, healthy tissue.
Yeah, I agree. So I think that these are processes that are important and help to maintain tissue homeostasis. And just looking at it through the lens of telomere biology, not to say that there aren't important processes that drive the aging process that you've mentioned, and I agree. I think that there also are opportunities to ask whether or not there are interactions between those processes.
Yes.
And inflammation, which is particularly ROS generating, is an
Right.
example, or the injury repair, where you'd have more cell turnover. Think about Crohn's disease, inflammatory bowel disease.
Right.
You have the opportunity to create more damage on telomeres and so on, right? So you can think about these things being quite interactive and actually fueling on each other. And we had shown actually in liver cirrhosis,
Yes.
when you engineer mice, when you have mice with long telomeres and you injure the liver, you don't get cirrhotic livers. As soon as you make telomeres limiting, you get florid stage 4 cirrhosis. And in that setting, what happens when you have limiting telomeres is that the apoptotic cells, the lipid peroxides from dying cell membranes, activate stellate cells that then cause this fibrotic reaction. So, you have this link between inflammatory process where DNA damage could create inflammation and vice versa, where inflammation
Right.
can create a high ROS state.
Thank you.
Have you had the opportunity to investigate telomere and protection proteins? And in your opinion, do you think they might play a role in future translational work?
We haven't, and I think that the shelterin complex plays a very important role in maintaining telomere biology, extremely important. I think most people simply think about telomeres in terms of attrition. That's not the case. It's really about maintaining the integrity of the telomere as a cap, which could relate to the amount of damage, could relate to the regulation of the T-loop as it relates to the shelterin complex. All of those things could be modulated in aging and in responses to telomere dysfunction that could be just as important as attrition. But we haven't looked.
Yeah, that's a very good talk. And I just wondering, in mammalian mitochondria, there's about 15 protein is contained, 1,500 protein in the mammalian mitochondria. 95% of this mitochondrial protein are encoded by nuclear gene. So I'm wondering if you have you see the mitochondria profile of this mitochondria influx of protein which is encoded by nuclear?
Well, certainly the proteins, and we looked at only a few by Western blotting. Certainly the proteins that are encoded in the nucleus, those are the vast majority that we saw that were repressed at the transcriptomic level, and where we looked, protein was also down. So the nuclear encoded network that PGCs regulate is down across the board. Now, we didn't catalog every single protein, and I have to say that I didn't carefully look at the list of all proteins for mitochondria, be it PGC dependent or independent. We haven't looked at that.
Well, thank you. Just like to let everyone know we have a chance to speak with Ron further if you'll join us for a reception, which you may remember is out this way in the library. So again, thank you, Ron. Look forward to a chance to talk with you informally. Also, just kicking myself for all the time I had with you, didn't get a chance to ask for your guidance in some of our own lab work. So
Yeah,